Right, next we're going to look at polyribosomes. Okay, so polyribosomes is when many ribosomes are translating one mRNA simultaneously. So you see a chain of ribosomes bound onto the same mRNA. Right, so this is done so that the translation process is more efficient. Right, there can be many copies of the same polypeptide made from the same mRNA simultaneously. Right, so this is how it looks like under the electron micrograph and cartoon to simplify the diagram. Right, so you see a chain of ribosomes. Right, all these black dots are ribosomes. Okay, and then they are all bound onto the mRNA. Right, so and you can also see the growing polypeptide chains. Right, it's shorter over here, it's slightly longer over here, so that tells you that the 5' is here and the 3' is here. Right, so as the ribosomes move down 5' to 3', the polypeptide is elongated. Right, so shorter here at the start and longer here. Right, okay, so this is basically a clearer cartoon representation of this diagram here. Okay, the polyribosome. Right, so these polyribosomes is found in both eukaryote and prokaryote. Right, both types of cells have such polyribosomes. Right, so many ribosomes can bind to the same mRNA to translate the same mRNA simultaneously. Right. Then we look at another type of diagram. Right, I'll go back to the previous slide later. Let me just look at this first. Right, so if you see this kind of diagram, right, what you see is this is the polyribosome that we looked at just now, right? A chain of ribosomes. Okay, you don't see the polypeptide, but you do see the ribosome. Right, again another one, right? Polyribosome here, a chain of new ribosomes on the same mRNA. Then what is this then, right? This line here. This line would be the DNA. Okay, so from the DNA we have transcription and we have the mRNA, and the mRNA is already covered with ribosome, even though it is still elongating, as you can see. Right, so transcription should occur right in this direction. Right, the mRNA polymerase, not mRNA, the RNA polymerase, which we cannot see that, would move along the DNA in this direction. Right, so this is the start of transcription. So the mRNA is shorter. This is the towards the end when the mRNA is longer. Right, and before the completion of transcription, translation actually started. And this is only possible in the prokaryote. So when you see such diagram, like this diagram with the polyribosome, as well as the DNA showing you the process of transcription. So transcription and translation are happening simultaneously. Right, this is only possible in a prokaryote. Right, so I'll go back to the previous slide to explain. The reason why is because in the prokaryote there's no nucleus. Right, transcription and translation, right, both occurs kind of in the cytoplasm. Right, so transcription occurs in the nucleoid region, which is within the cytoplasm, and it is not separated from the rest of the cytoplasm by a nuclear envelope. Right, so even before the transcription is completed, whatever mRNA that has already been synthesized, as long as there's the shine-diagonal sequence, the ribosome can bind. Right, as long as there's a shine-diagonal sequence, the ribosome can bind. Right, so even before transcription is completed, translation can already be initiated and translation can happen. Right, so simultaneously, both transcription and translation occur. Right, so this is the polyribosome that you see, and it is bound to the DNA because transcription is still occurring, just like this diagram over here. All right, but for a eukaryote, there is the separation of transcription and translation because of the presence of the nucleus or the nuclear envelope. Where transcription occurs in the nucleus, translation cannot occur because the ribosomes are not in the nucleus. The ribosomes are in the cytoplasm. So even though during transcription, right, after after transcription has started, there might already be the 5' UTR, but there are no ribosomes in the nucleus to bind to the 5' UTR. Right? So translation cannot initiate, right? So translation can only begin when transcription is completed in the nucleus and when post-transcriptional modification has occurred in the nucleus. Before the mature mRNA leaves the nucleus and enters the cytoplasm, only then can translation be initiated. But so in the eukaryote, you don't see the simultaneous transcription and translation like the prokaryote. You only see the polyribosome that we look at here, right? This you can see in the eukaryote without the DNA, right? But these, right, the polyribosome with the DNA are only seen in the prokaryote. So when you see this, you know this is prokaryote. The question don't have to tell you, right? The question doesn't have to tell you. You know this is prokaryote, right? So that's the polyribosome part.